In this video, we'll be looking at solutions and some of the qualitative things that happen with solutions. So the first thing we want to talk about when we're dealing with solutions are the ability to act as electrolytes. And what an electrolyte is an electrolyte means something has the ability to conduct an electric current when dissolved in a solution. When we think about pure water is not a very good electrolyte. It does not conduct an electric current very well at all. It's the things that can be dissolved in water that can potentially conduct an electric current, which is why you may have heard the phrase, water and electricity don't mix. It's not so much that it's the water and the electricity that don't mix, it's the things dissolved in the water that can cause problems with conducting an electric current through that solution. So that's why you don't often do plumbing and electrical work at the same time. Not so much because the water conducts the electricity, but because the things dissolved in the water are capable of having that solution conduct electricity. There's three types of solutes that we want to talk about based upon how they act as electrolytes. And for this, the names are kind of hinting at what they're going to do. We have strong electrolytes, non-electrolytes, and then weak electrolytes. So we're going to talk about each one of those individually, but just by the names, they're hinting at what are their abilities as electrolytes. The first type of electrolyte we want to talk about are the strong electrolytes. Strong electrolytes will readily conduct an electric current. And the way they're able to readily conduct an electric current is they produce many ions. And what we're going to see is that it's the ions in the solution that give that solution the ability to conduct the electric current. So when you're trying to figure out something is a strong electrolyte, think back to things we've already talked about. What's going to produce a lot of ions? Well, ionic compounds are those that are going to produce a lot of ions. Here we're seeing an example of a strong electrolyte. An example of a strong electrolyte is calcium chloride. Calcium chloride is a solid. That's what the little S represents in parentheses after the formula. Calcium chloride, you may be familiar with, is sometimes used as a de-icing agent for driveways, sidewalks. Calcium chloride can dissolve in water, but when it dissolves in water, as it dissolves, the water molecules are completely surrounding each ion, such that we're really having free calcium ions and free chloride ions. And what you often see when we have things dissolved in water, water is just such a common solvent. We're dealing with solutions that are water-based all the time. This AQ after. The AQ is just our abbreviation for aqueous, and that just means we are dissolved in water. So you can see for every unit of calcium chloride, we're producing a lot of units of ions. Each unit of calcium chloride produces 3 units of ions. 1 calcium ion and 2 chloride ions. So if you had 100 units of calcium chloride, you'd get 300 total units of ions. 100 calcium ions and 200 chloride ions. So you want to associate your strong electrolytes with your ionic compounds. On the other extreme are the non-electrolytes. These, just like the name is hinting at, do not conduct an electric current. And the reason that they do not conduct an electric current is these produce no ions. We want to think about these as our covalent or molecular compounds. A good example of a non-electrolyte that you're probably familiar with is glucose. Here we have the formula of glucose, C6H12O6. Right away, this should be screaming that this is a molecular compound. This is a covalent compound. It's not going to produce any ions. You see all nonmetals in the formula, carbon, hydrogen, oxygen. You don't see any polyatomic ions. You don't see anything that's going to say, hey, we're dealing with an ionic compound. This one's definitely molecular. We know that glucose will dissolve in water. After all, glucose is dissolved in our blood, so blood is mostly water, so we know that glucose will definitely dissolve in water, but when it dissolves, it's dissolving as whole units. So it still dissolves. The glucose units are still surrounded by water. You still get a homogeneous solution, but none break up into ions because it isn't an ionic compound. So you don't produce any ions from these non-electrolytes. So even if you had 100 units of glucose, you would produce 0 units of ions. You just don't get any ions coming out of these non-electrolytes. Our last type, our weak electrolytes, are kind of in the in-between ground between the strong electrolytes and the non-electrolytes. These will partially conduct an electric current or weakly conduct an electric current. We saw what happened when we had a lot of ions. That was a strong electrolyte, a great conductor of an electric current. We saw what happened when we had no ions. We had a non-electrolyte not able to conduct. So these weak electrolytes will be in the middle, will produce a few ions. These can be very difficult to identify just by looking at the formula. Generally, you have to be given some sort of hint or some sort of idea that you're dealing with a weak electrolyte. So let's look at how these weak electrolytes respond. So an example of a weak electrolyte is HF. And HF is actually a gas. That's what the little G represents. And when you place HF into water, the first thing that happens is it dissolves as whole units. Then a secondary thing can happen. And the secondary thing is that some of those HF molecules will split up into ions, but it's only a small amount. And the way you can recognize that it's only a small amount is this with this double-headed arrow. You see, in this case, we have a double-headed arrow. That means both processes are happening, both forward and reverse. And sometimes you'll even see the arrow with only a very small one written forward and a much larger one written going in the reverse direction, kind of further illustrating that not very many of these particles are actually going to break up into ions. Usually for these weak electrolytes, over 95% of the particles stay together as whole units, and less than 5% actually break up into ions. So if you started with 100 units of HF, you may only get 2 H pluses and 2 F minuses. You may only get 4 units of ions coming out of that if only 2% of these break up. So you get a little bit of ions. Remember, for the non-electrolytes, we got absolutely no ions. For the strong electrolytes, remember when we had 100 units of that strong electrolyte, we were producing 200-300 units of ions. Here, starting with 100 units of HF, we're only producing 4, 5, 6, 7, 8, somewhere around there, units of ions. So we only get a small amount of ions from these weak electrolytes, but it's enough to partially conduct that electric current. So these are tough to identify. You really only have 3 ways of identifying these. One is if you're just outright told, hey, this molecule is a weak electrolyte. That'd be the first way. The second way you might be able to identify a weak electrolyte is if just kind of using words they describe, hey, this one will partially break up into ions, or this will only produce a few ions. And then the third way is if you see an equation, you can figure it out based upon that double-headed arrow. All the other ones fully produce ions, like the strong electrolytes. That shows you you're making all of the ions possible out of that. These weak electrolytes, you only produce a very small amount of ions from. Next thing we want to talk about is, besides solutions, there's a couple other types of liquid mixtures that we want to briefly talk about and discuss. So we're going to do this kind of in a table format. So there's 3 different types of liquid mixtures. There are solutions, colloids, and suspensions. And as we go through and we talk about these, you want to think about a key example of each, something that you're familiar with, because that's going to help you understand these various properties that each one of these mixtures have. So common solutions that you're probably familiar with, salt water or sugar water is a good example of solutions, as we've talked about before. A good example of a colloid is milk. And for suspension, orange juice with pulp is a good example of a suspension, or coffee with the grounds still in it would also be a good example of a suspension. Now, the most important column in this chart is the particle size, because it's the particle size that differentiates whether you're going to get a solution, a colloid, or a suspension coming out. So in a solution, the particle size is relatively small to medium-sized particles. And that's going to be very important in terms of those properties. So we want to look at those properties. Because the particle sizes are not very large in a solution, will light be able to penetrate it? Yes. If light can penetrate that liquid mixture, then it's going to be clear. Doesn't mean it will have to be colorless. It can still have color to it, but it will be clear. Because the particle sizes are not very large, will those particles settle out if you just leave it sit? And the answer is no. Because the particle sizes are so small, just the motion of the solvent particles will keep those solute particles mixed up in the solution. So they will not settle out. Last thing we want to look at is how can we actually separate these, or separate parts of the solute from the rest of the liquid mixture. And for this, for solutions, we can think about two different ways we could maybe separate something. That's either by membranes, sometimes called semi-permeable membranes, and filters. Solutions, because the particle sizes are so small, they can't be separated by either membranes or filters. You can think about a membrane as a filter just on a super small scale. So because solutions have such small particle size, light can penetrate them, they'll be clear. The components are not going to settle out, just the molecular motion will keep everything mixed together. And it's very hard to separate them either by membranes or filters. When we go up to a colloid, something like milk, the particle size in colloids is very large. Some of the components in milk, some of the proteins, some of the sugars, some of the fats, unless it's skim milk, are very large. That larger particle size is going to lead to different properties than we saw for the smaller particle size in solutions. When you have a larger particle size in a colloid, light's not going to be able to penetrate it that easily. The particles are large enough to block the light from passing through. That means they're going to be opaque. Will the particles settle out? They still won't settle out. You can look at your regular gallon of milk or half gallon of milk, and it doesn't really tell you to shake before use because, well, there's no reason to, because nothing's going to settle out of that. Even though the particles are larger, there's still enough molecular motion of the particles to keep those thoroughly mixed together. For separation, filters aren't going to work, so these cannot be separated by regular filters. Try pouring milk through a coffee filter. It's going to go right through. You're not going to separate anything out. But if we use semi-permeable membranes are actually small enough in order to separate out some of those large particles in that colloid. So semi-permeable membrane works just like a filter, but on a much smaller level. The bottom right-hand side of the screen, you see a little bit of a picture of what a semi-permeable membrane looks like. The dashed vertical gray line represents that membrane. And that membrane has gaps in it that will trap some particles. You can see the very large green particles, the green balls, cannot pass through that membrane. They're going to get blocked by that membrane. But the much smaller blue dots, they're not going to be able to be trapped by the membrane. They can go in and out. So because colloids, some of the components in the colloid are large enough, example kind of being those green balls you see in that bottom right hand picture, they can be trapped and they can be separated by using these semi-permeable membranes. The last type of liquid mixture is a suspension. These have extremely large particle size. So the particle size here is very, very large. They are so large that usually you can see the particles with your naked eye. You know, think about it. You can see the little bits of pulp in orange juice. You can see the grounds in coffee before it's been filtered. Think about a muddy water. You can see the little bits of dirt in there. So particle sizes are so large, light won't penetrate. If light can't penetrate, it will be opaque. Here, the particles are so large that eventually they will settle out. And you have some experience with this. Think about mopping a floor. That dirty water that you've mopped the floor with, if you let that sit for a while before dumping it out, you can actually start to see some of that dirt settle out to the bottom. You're also familiar with this when you look at a pond, a lake, a river, or a stream. After a rainfall, when all the dirt and mud has rinsed into that pond or that lake, it's very, very turbid. It's very, very murky. But if it sits for a while, you'll start to clear up because those large particles are starting to settle out. These particles are so large that they can be separated by filters. We use filters for our coffee. You're familiar with how they work. The particles are large enough that they're going to be caught by those filters, and you can separate them. So when we're looking at our different types of liquid mixtures, the real properties are all based upon particle size. The light penetration, will they settle, how they're going to be separated, if they can be separated easily, is all based upon the particle size. So that's something you're going to want to kind of really pay attention to, because that's what really differentiates solutions, colloids, and suspensions, is how big are those particles. The next thing we want to talk about is relating different types of solution in relationship to a cell. And this is based upon concentration. And we're not really doing calculations with this, but there's some terms we want to understand, and we want to see what happens to a living cell when they're exposed to these three different types of solution. The first type of solution we want to talk about is a hypertonic solution. In a hypertonic solution, that's a solution that has a greater solute concentration than what you would find in a cell. You have more solute of that particular type in the solution than you would normally find inside of a cell. You can also have the exact opposite. The exact opposite would be called a hypotonic solution. Here, the solution has a lower solute concentration than what you would typically find inside of a cell. You also have what's called an isotonic solution. In an isotonic solution, that solution has the same solute concentration than what you would usually find within a cell. So we want to look at what's going to happen to a cell as we place cells in these different types of solutions. The first type of solution we want to look at is a hypertonic solution. So we can see in our diagram on the left, the large circle represents a cell, and in our diagrams, X's are going to represent the solute particles, and the blue O's are going to represent water molecules. And we can see this is definitely a hypertonic solution. We have a much higher concentration of solute particles outside the cell than we do inside the cell. So whenever we have a difference in concentration, something always tries to happen. Whenever we have a difference in concentration between two things, it's going to want to try to equilibrate. And it's going to want to try to equilibrate by going from regions of higher concentration to regions of lower concentration. And you have some experience with that. You can think about food coloring. If you put a drop of food coloring into water, that drop of food coloring is very, very concentrated in that food coloring. The water that you're placing in doesn't have any food coloring in it at all. It's not concentrated at all. It's very much dilute. What happens is you see the solute particles starting to disperse throughout the water. They're going from where they're very, very highly concentrated in that drop into the rest of the solution. Another way of thinking about it is the water molecules are very, very concentrated in the pure water, not as concentrated in the drop of food coloring. The water particles are also going from regions of high concentration to regions of lower concentration to mix with that dye. So we have that experience with food colorings and dyes of things wanting to equilibrate. If they're concentrated, they're going to want to spread out and kind of equilibrate to thoroughly mix together so you get equal concentrations throughout. The same thing wants to happen here, but there's something that's preventing that. We have more solute particles outside of the cell than inside of the cell. So in order to equilibrate, these particles are going to want to try to get into the cell, but there's something that's preventing that. What I'm highlighting right now is the cell membrane. The cell membrane prevents that free passage of these solute particles. So these solute particles can't equilibrate. The cell membrane is blocking them. But what has free passage inside and outside of the cell through the cell membrane, what the cell membrane doesn't block, is water. If we look at the water molecules in here, we have much more water, a higher concentration of water inside the cell than outside the cell. Water molecules can pass freely inside and outside of the cell. So some water molecules are going to leave the cell, others might come in. Ones may leave, others may come in. But because I have much higher concentration of water inside the cell than outside the cell, in order to try to equilibrate, more water molecules are going to leave the cell than enter the cell. What does that mean? We're going to get a net loss of water from the cell because more water molecules will leave than enter, trying to equilibrate the concentrations of the water. We think about the major components in a cell. The major components in a cell is water. If you start losing water, you're going to really alter the shape and size of that cell. And we have a term for this when we have a net loss of water from the cell, and that term is crenation. So crenation is the shrinking and shriveling of a cell from that net loss of water. You can think about this the same way you would a water balloon. You let some water out of a water balloon, that water balloon gets smaller. Same type of thing is happening in a cell. So when you place a cell in a hypertonic solution, you're going to get a net loss of water. Crenation will happen to that cell as it is losing water from being exposed to this hypertonic solution. In a hypotonic solution, we have almost the exact opposite thing happening. Here we have a diagram of a hypotonic solution. Remember, in a hypotonic solution, you have more solute inside the cell than outside. In the solution, you have less solute. So you can see that in the diagram. I only have one solute particle outside the cell. I still have my 3 solute particles inside the cell. So again, they're going to want to try to equilibrate, but the cell membrane is going to prevent that. The solute particles don't have that free passage into and out of the cell. but the water molecules do. So water molecules can enter the cell, they can leave the cell. Some will come in, some will go out. Some will come in, some will go out. But because we have a much higher concentration of water outside the cell than inside the cell, we're going to have more water enter the cell than will leave the cell. So we get a net gain of water by that cell. So if we get a net gain for water on that cell, what might happen to that cell would be lysis. So what lysis is the process of the cell expanding and then bursting from that net gain of water. You can get so much water being taken up by the cell that the cell membrane can't withstand the pressure from within it anymore, and it can actually rupture and burst. Again, this works the same way as a water balloon. If you overfill a water balloon, it's going to burst when the pressure inside can't be contained anymore. The same thing can happen inside of a cell. So for hypotonic solutions, if you place a cell in a hypotonic solution, you're going to get a net gain of water. As that cell starts gaining more and more water, it's going to expand. It could eventually get to the point of not being able to withstand the pressure of the water within it, and it can rupture and burst. We call that process lysis. Our last type of solution is an isotonic solution. In an isotonic solution, remember, we have the same concentration of solute inside and outside the cell. That means we'd have the same concentration of water inside and outside of the cell. This is still a dynamic process. Things will still happen. Water will still enter the cell. Water will still leave the cell. Water will enter. Water will leave. Water will enter. Water will leave. But the same amount that enters will be the same amount that's leaving. So if the same amount is leaving that is entering, there will be no net change in the water content for the cell. Water is still going to enter and leave. It's just the same amount that's coming in is the same amount that's going out. So there's going to be no net change in what's happening inside that cell. I'll talk just a little bit about the idea of solubility. What solubility means is how much solute can dissolve in the solvent. And you may have heard the expression like dissolves like. What like dissolves like means is that polar solutes dissolve more easily in polar solvents. Non-polar solutes dissolve more easily in non-polar solvents. You have a matching. Two things that are polar are going to mix well. Something like salt dissolving in water. Water is polar, salt is ionic. That's like the extreme of polarity. They dissolve in each other very, very well because they're both polar. You know that oil and water don't mix. Well, oil and water don't mix because the water is very polar. Meanwhile, the oil is very, very non-polar. So those are not able to mix and dissolve in each other. So you're kind of familiar with some of this idea of like dissolves like. A couple of terms that get used that you want to be familiar with: the idea of a saturated solution. A saturated solution is one that contains the maximum amount of solute that can be dissolved. If you're at the maximum, you can't dissolve anymore. No more solute can dissolve. You can compare that to an unsaturated solution. An unsaturated solution, not saturated, this is a solution that contains less than the maximum amount of solute. More solute can be dissolved in here. So some terms we want to be familiar with: saturated, unsaturated. We should be familiar with the idea that like dissolves like. Two polar things will mix well and dissolve in each other. Two non-polar things will mix well and dissolve in each other. But polar and non-polar are not going to dissolve in each other. I'm going to talk a little bit about how temperature ends up affecting solubility. And it matters what type of solute you are. If you are a solid solute, what will happen is you will have an increased solubility as temperature increases. And the reason this happens is the additional energy at higher temperatures gives you more molecular motion. The higher the temperature, the more energy, the more motion. It makes it easier for the solvent to surround the solute particles and dissolve it. Example that you're familiar with, you can dissolve more sugar in hot water than you can in cold water, because that extra temperature gives you increased molecular motion. You can dissolve more solute in it. If your solute is a gas, the exact opposite happens. For a gas solute, solutes that are gases will have a decreased solubility as temperature increases. The additional energy at the higher temperatures give the gas particles more energy. Well, for a gas, they can escape that liquid barrier. They can escape out of that solution because they're gases. They have more energy. They're moving faster. It's easier for them to escape the surface of the liquid. Once they escape the surface of the liquid, they're gone. They're not going back in there. You don't have that problem when we talked about solid solutes. A solid solute doesn't have a way of escaping that solution. Gas solutes do. So as you increase the temperature for a gas solute, you're giving it more energy. It's having an easier time breaking through that surface of the liquid and escaping. And you're familiar with this. These are things that you already knew. Just to give an example, think about a carbonated beverage. A warm carbonated beverage will go flatter faster than a cold carbonated beverage. If you're unsure of this, you can run the experiment for yourself. Take 2 carbonated beverages, one cold, one warm. Open up the warm one, leave it on the counter. Open up the cold one, leave it in the refrigerator. Come back in a couple hours. See which one has more carbonation. The colder one will have more carbonation because it was able to retain more of those CO2 gas particles in order to stay carbonated. So these are things you're already familiar with. You're familiar with dissolving more sugar in hot water than cold water. You're familiar with warm carbonated beverages going flatter faster than a cold carbonated beverage. These are just real life examples of solid solutes increasing solubility as temperature increases, and gas solutes showing a decreased solubility as temperature increases.